We know that there's excessive band activity that really relates to degree of cognitive impairment and severity of anxiety. And with a single dose of the Spinogenix drug in Fragile X, that completely normalized. That's the strongest effect we've ever seen. So if we can keep that up, that's then a brain that is just able to process information differently.
Welcome to Power to the Patients. I'm Brandon Lee, and today I'm honored to have Dr. Craig Erickson with us today. He's a leading expert in neurodevelopment disorders. He's also professor of psychiatry at the University of Cincinnati College of Medicine. Craig leads a pioneering program at Cincinnati's Children's Hospital that focuses on understanding how treatments engage the brain at the synapse level, enabling therapies to be tailored to individual patients from the earliest stages of clinical trials. He's also the director of the Cincinnati Fragile X Research and Treatment Center, and the inventor and holder of multiple patents across Alzheimer's, ALS, Fragile X syndrome, schizophrenia, and more. At Spinogenix, Craig is helping to develop innovative drugs that aim to restore synapses, a fundamental building block of the brain. And today we're going to dive into the importance of synapses and the work that Spinogenix is doing. Craig, welcome to the show.
Oh, thank you so much for having me. Really excited about the discussion.
Yeah. Let me just lob a high-level question your way to start. Why synapses?
Well, I think from a brain functional standpoint, we have thousands and thousands, millions of brain cells, neurons that need to connect. And it's those connections between cells that really form a common thread across a number of brain disorders. So neurodegenerative disorders, things like ALS and Alzheimer's, significant severe mental illness like schizophrenia, and neurodevelopmental disorders. So we see a lot of common threads that signaling from cell to cell at these synapses that are really connections. And they have both what's called a pre and a post-synaptic component. So the sort of the cell that's trying to get the message down the chain in a way. And so that chain, those links tend to break down in different ways, leading to a lot of different brain pathology. Again, as broad as from ALS and Alzheimer's to I study Fragile X, which is a genetic cause of autism. A common link is this synaptic or these connections that degenerate and break down for different biologic reasons. And if we can restore those connections, we can really have a more functional brain.
Yeah. I've had a bunch of conversations on this show across psychiatry and neurology, and rarely do I have conversations where folks have applications that seem to span neurodegenerative, like autism, as well as psychiatry, like schizophrenia. Can you talk about how these are different, but then also similar in the way that maybe synapses play a role in potentially treating those kind of conditions?
Yeah, I mean, I think that the synapse becomes kind of a common thread, but there are many different ways that various brain pathology can cause that damage. And also at different times of your life. So, for example, Fragile X syndrome, a single gene cause of autism that I study, is there and having a brain effect from the fetal time all along throughout development. Something like, Alzheimer's and ALS, a more classic adult onset, schizophrenia, often young adults, but we see these common end results of a brain that just can't functionally connect. But the manner in which the pathology breaks those connections down is unique. It's unique in its development. It can be unique in specific parts of the brain that can have different more cognitive or more behavioral or more motor effects. And so there's a lot of individual nature of the patterns that synapses could break down that can have these different pathological endpoints. And it also matters when that pathology develops, from birth and early, like with these childhood developmental disorders, versus later in adult onset, and where the patterns different. But the commonality, it's with the Spinogenix treatment approaches across, as you said, these diverse areas of brain pathology, is the synapse, is the connection. And if we can improve that connectivity, do we have potential impact across broad disorders in ways that you're right, you just don't hear about? Being able to target these very diverse conditions with a similar general approach.
I imagine that there are many different ways in which your career could have gone. how did you get obsessed with synapses as a
Well,
path?
I got originally obsessed with neurodevelopmental disorders. So I actually, before I went to medical school, I worked summers at camps for kids with autism and related disorders. So I wanted to be a physician, but I had firsthand experience working with youth and young adults that had severe developmental disabilities. And I was just struck by the degree to which their communication and their cognitive deficits impacted their day to day life, requiring adult supervision throughout life. So that got me into being a brain scientist and a neurodevelopmental expert. From there, I really followed the research that was coming out, a lot of stuff into the 90s, into the early 2000s, which was the formative time of my career and medical training and residency and fellowship, pointing to the synapse, pointing to aberrant signaling via neurotransmitters, aberrancy in protein synthesis at the synapses. And that's what really brought me through to that conclusion that, hey, there are a number of therapeutic synaptic targets that affect neurotransmission, protein synthesis, the actual morphology of the synapses, which Spinogenix, lead compound in Alzheimer's and ALS and schizophrenia, changes the morphology. Actually, the synapse is you're able to get mature synapses. I never thought that was possible, but that's how I originally got into this, following the science originally from just personal experience around individuals with severe developmental disorders.
Wow, it's amazing to hear the kind of arc of your career and the kind of like intentional path that you've taken to kind of work with these groups. Since we're starting to get there, I'd love to hear more about Spinogenix. What's the story? How did it come to be?
Well, my connection with Spinogenix is with their chief scientific officer, Pete Vanderklish, who is a scientist I've known from Scripps and the San Diego area for years because of his work with synaptic pathophysiology and synaptic science in neurodevelopmental disorders, in Fragile X and in autism. And so we met, and he connected me with Spinogenix and both their work in Fragile X, which is with a large conductance BK potassium channel modulator, and their SPG 302 work in Alzheimer's and ALS and schizophrenia, because I knew about all of his work with synaptic preclinical science. And I was able to bring to Spinogenix not only that interest in synaptic pathophysiology and treatment development, but my NIH funded portfolio in the last decade plus has been really focused on using EEG and other measures to look at brain target engagement in early phase studies. So, in other words, we care about clinical outcomes, but if we're doing first in disease disorder studies and we have small number of patients, how can we quantitatively show if brain function is normalizing from an electrophysiologic EEG signal standpoint? And Spinogenix was very interested in taking everything we've learned from our federal research using EEG and other measures in early clinical trials and bringing it into their early phase trials. So it was a nice match there of the synaptic science, but then this interest in highly quantitative trials, which is borne out really well for Spinogenix with their early work, because there's no placebo response really with whether your brain is oscillating more normally at high or low frequencies. It either is or isn't happening, and that's a robust tool for first in disease studies.
Yeah, especially in a therapeutic area that is so burdened sometimes with like subjective readings. Can you tell me a little bit more about the tags platform? As I start to learn more about it, I'm starting to see how it's spawned out SPG 302 and SPG 601. I'd love to just understand a little bit more about the platform as a starting point.
Yeah, I think the platform is really, a novel small molecule way to, again, enable neurons to essentially generate or regenerate mature synapses where they haven't existed. It's that kind of microscopic anatomical change that's very unique. I've done a lot of work with neurotransmitter drugs, right? Like SSRIs and dopaminergic drugs and glutamate and GABA modulators. But the tags approach itself is really more at this, microscopic anatomy changing brain connections. So changing the morphology via the proprietary change in how brain cells are able to remodel, which is super novel. And then the 601 in Fragile X, the large conductance potassium channel modulator, is more of a traditional neurotransmission modulatory approach, but also restores synaptic signaling and connectivity. So they share the synapse, but they're a little bit different at the core. And my real focus in Fragile X expertise in 601 is, can we take the 601 compound, the potassium channel modulator, do a small study in a rare disease and detect signal? And the result is we have, which just in 10 men with Fragile X has been really momentous, but it's because Spinogenix was willing to commit to a really quantitative, like, what is patient performance like on drug? What is the EEG signal change on drug? And a lot of pharma don't take that approach. They just go after hundreds of patients and look for small, subgroups based on lots of clinical data with post hoc analyses. But that's not the approach we've taken.
Yeah, can you tell me a little bit more about like the from to observation that you see, and like how that's maybe different from like the, let's call it, traditional way of doing things?
Well, I think the traditional way of doing things, is very kind of large scale. So you're going to go into Fragile X or autism or ALS or Alzheimer's, and you're going to dose a whole lot of patients and kind of hem and haw about, are we seeing clinical change? You get a lot of placebo effect. You kind of end up, and we've done this a lot in neurodevelopmental disorders. You catch yourself with post hoc comparisons where you're like, well, we thought we were using this scale, but this tertiary or secondary measure looks like it's changing in a subgroup, and you end up kind of chasing these things. And the different approach from sort of beginning to end with Spinogenix is, it's like, if we're going to enter a disease and a disorder, we're going to look at, for example, what we did in Fragile X. We did computer-based performance testing, and we did high-density EEG study to see what brain electrical activity change was happening, if it rescued known abnormalities in the disorder, and if in parallel, patient performance on cognitive testing improved. And in ALS, for example, there's bringing in EEG and transcranial magnetic stimulation as exemplars to look at brain functional change along with more traditional observational measures. That's just a different approach of being able to make go or no go decisions based on highly quantitative data. It enables you to do studies very quickly, burden a smaller number of patients, and then sort of launch from there.
Yeah, if I may be difficult for a moment, this sounds ideal. Why doesn't everyone else do it this way, using EEGs and these more objective markers?
It's a great question. When I got into this work 10 or 12 years ago with objective markers, I kind of scratched my head too, but I had a career of negative trials. I mean, I like to say I've been involved, for example, in autism with every negative, double-blind, placebo-controlled, multi-site, large-scale trial. We just failed after failed after failure, and that made me reconsider our approach, because I would take treatments that in clinic, open label treating, off label use of medicine, 2/3 of patients in clinic would seem to be, quote, improved, whatever that meant. And then we would enroll 100 or 200 kids with autism as an example, and we would never beat placebo. And we would never have quantitative measures of what the drug was doing to affect an individual. So we shifted, and I think it's not a typical way for pharma to approach a lot of new indications, and I think it really is sort of a state of the art, newer de-risking strategy of future larger phase trials to make go or no go decisions. Right? I mean, that's really what it is. And with the 10 men in Fragile X that we enrolled in the Spinogenix trial here at Cincinnati Children's with Fragile X syndrome, if we would have seen absolutely nothing, I would have said, look, this may not make sense to move forward, but can we figure this out in 6 months versus 2 years in 10 men versus 100 patients? And I think the answer is yes. So I think we're trying to change sort of the tone and approach in neuroscience trials to de-risk things. And I only came out of that through massive failures. Almost 2 decades of failed trials for core symptoms of autism. We have to do things better. So I really moved to how can we use biologic markers of brain function that are quantitative and placebo resistant? And I think that has legs. We're seeing a lot more interest in it, in using quantitative EEG and an analysis type we call source localization, that you just get a lot more usable data from small samples. But the proof will be in the future, right? Like it'll be in, can these informed go or no go decisions result in approval? So that is still to be shown. To your point, Brandon, we need to show that. But I think the early returns are, we can at very least discern this is showing good target engagement, this is doing absolutely nothing, and help the directions in the future decisions.
Yeah, it's fascinating. I've had Dr. Amit Etkin from Alta on the show, who's also using EEGs across a variety of different psychiatric programs. And one of the things that we spoke about was whether the use of these markers is viable as an endpoint, or more applicable at later stage, more as an entry criteria or an eligibility criteria. I'm curious, how is that evolving on your end, and how are you seeing that evolve?
It's a great question. It's stuff I talk to the FDA about in many
I
different
bet.
settings. I interface with the psychiatry branch, mainly at the FDA. And, the FDA biomarker qualification process for an approvable endpoint is onerous, right? And that's the only way to describe it, and takes a ton of time. I really see EEG and computer-based testing and transcranial magnetic stimulation as a biomarker as outcomes in the earliest phase studies to justify further work, and, as you said, as inclusion stratification criteria. For example, in the Fragile X work with Spinogenix, we saw that the SPG 601 drug had this clear EEG signal effect on high and low frequency power that I know is really pathognomonic of males that make zero Fragile X protein. So not only males with Fragile X, but the specific 2/3 of the men that have essentially no protein expression. We know they have this EEG phenotype that's being normalized. So then I can make that link to study inclusion criteria. I don't even necessarily have to use EEG because we have it so well established in Fragile X, linking the molecular phenotype to the EEG phenotype. But in other areas like autism, absolutely, we can use EEG entry criteria. And the feedback I get from the FDA is, hey, we're fine for you to enrich your sample in a biologically a priori logical manner of who you think may respond best. But we're not going to give you a new drug approval based on an EEG marker. And so that's how I envision it, both de-risking moving to the larger scale studies, but also as thinking about inclusion and being able to parse populations, which can be very useful, especially in something as heterogeneous as autism, for example.
Yeah. You know, one of the frustrations I've heard, over and over again, working with folks doing neuroscience and psychiatry research, is like the fact that these objective markers are not yet established as endpoints. And I hear a lot of, oh, it's onerous, it's onerous. Could you tell me why it's so onerous, and what's going on in those conversations when you advocate for it?
Yeah, I mean, I think, the blueprint, if you look at the federal guidance for biomarker approvable quantification, is really years of validation studies linking the quantitative measures to clinical outcomes, not only in cross-section, but also over time. So it's just difficult. In my career, I noticed industry developing even, their own new pen and paper measure, and getting that through qualification with the FDA, and seeing the hundreds and hundreds of patients of data that's been needed, and seeing the years that it's taken. And that's cheaper and more simple than qualifying a biologic electrophysiologic marker, for example. So there's a really, really high bar there. And we'd like to see it give a little bit. One win we've had, so we kind of talk about the negative, but a win we've had is in Fragile X, a rare disease affects 1 in 4,000 people. The FDA has become open to using a quantitative computer-based test called the NIH Toolbox as a primary outcome. So in
Hmm.
our Spinogenix SPG 601 trial, we used the toolbox along with EEG, and we actually had a statistically significant improvement on part of that computer-based test that could be moved forward as an approvable endpoint. So that's nice. There's some give there, but that came out of years of NIH funding of developing the toolbox as an outcome in Fragile X and autism and other developmental disorders. So there are some positives there, but it is difficult. But I'm hopeful about that as a performance-based test as an outcome that we can take through phase 3, and it currently is right now in phase 3 in other Fragile X trials. So I think there has been a little bit of a give.
Yeah, I think it's fascinating and also really exciting to hear that we're making kind of this progress, not only with these outcomes, but also with EEGs, especially in Fragile X. Um, as you mentioned, you've seen the last 2 decades of work here and attempts here, maybe largely falling apart versus placebo. I'm curious, like, are there any of the kind of attempts over your kind of career that you'd want to run again with these new tools at your disposal?
That's a really great question. I was involved early in my career with a lot of glutamatergic and GABAergic modulators. For example, I did work with memantine, the drug FDA approved for Alzheimer's dementia, Namenda. We use that drug in autism, targeting core language and social impairment. And I'm a clinician scientist at heart, and we saw some individuals with autism who we gave them memantine and they communicated more. And I've published that, 15, 20 years ago, but we could never quantitatively define who they were. I think now if we had some of these more quantitative metrics like EEG, like expressive language sampling, computer-based testing, if we redid those large studies, could we quantitatively know the patient subpopulation that showed the overall clinical change? I think that's one example where we maybe could have done so. But it's you can't redo the past. All you can do is take the novel molecules of the present and learn from your mistakes. And that's what I'm hopeful. And the BK large conductance potassium channel modulator in Fragile X had a ton of preclinical validation going into it. We didn't know what we'd see in humans, but boy, we normalized EEG signal and we improved computer-based cognitive testing to a degree we've never seen before in an early phase Fragile X study. So I'm buoyed by the present and try not to regret too much from the past. But you're absolutely right. If we had the same tools in the last 20 years, we very well could have an approval in autism, for example.
Yeah. And certainly if it proves to be a pathway, I'm sure it would invite more kind of reconsideration along the way. You mentioned just like the heterogeneity of autism, and you've been speaking a lot about Fragile X. Could you tell me a little bit more about how it's different from other forms of autism and how you think about the different forms here?
Yeah. So, Fragile X is a single gene disorder. It results from a triplet repeat expansion in the FMR1 gene. And then what you get is reduction or silencing of Fragile X messenger ribonucleoprotein expression. What my lab and others have discovered and validated is it's not absolute. It's more of a continuous deficit. So it's also X-linked. So females with Fragile X are phenotypically infinitely variable due to random X inactivation. So I see hundreds of female patients, saw a bunch this morning already, and they're very variable. They may or may not have intellectual disability. They may or may not have autism. They tend to have anxiety, the majority of them. But males are more uniform. It's X-linked. They only have one X chromosome. So if their FMR1 gene is not functioning correctly, that's impacting every cell in their body. But even with the males, we learned that only a third of them make zero Fragile X protein. A third make trace about 5% of normal, and the other third are kind of in a 5 to 20% range. So that's heterogeneity, even in a single gene disorder. And because of that, when we did our first Spinogenix Fragile X trial, we only enrolled males with Fragile X that made zero or trace amounts of FMRP. again, because I knew they had a consistent EEG phenotype, and we could set up EEG then as a marker of target engagement. So if you have that degree of heterogeneity in a single gene disorder, then you look at all of autism, and it's infinitely heterogeneous. But we've had some autism success too, and it's led to the Spinogenix success. We work with a group called Stalicla out of Europe that did a sub-phenotyping approach to autism. They call it phenotype 1. We did a small 12-patient adult autism trial in this sub-phenotype. Again, EEG, cognitive outcomes, and they had an amazing result. And again, I think we have to be winnowing down these heterogeneous disorders in different ways. In Fragile X, it's we can do protein testing. In autism, Stalicla did machine learning and other approaches to having a more homogeneous phenotype. And then if we get a more homogeneous baseline phenotype, use all of our quantitative measures like we did in the Spinogenix trial, I think we have a chance of success. And then success is even a negative result because I think we have some assurance that we have reliable brain measures of efficacy.
I'm curious to hear, given the kind of current state of where you are in development and understanding Fragile X here, what's next? Like, how do you think about the next cards you want to turn over?
Well, I think, next with SPG 601 is phase 2B and phase 3 studies. I mean, it's poised as much or more than any molecule in the space till we need to do larger chronic dosing studies. I think there's a ton of excitement about that. And since Spinogenix, in-licensed a molecule that it already had some phase 2 safety data from work in multiple sclerosis, it really de-risked bringing it into large numbers of patients with neurodevelopmental disorders. So we're super excited about that. And I think the first focus would be on this male subpopulation that's the most homogeneous of the single gene domain. But then we would really look forward to separate studies, in females and potentially in mosaic males that are again making 5 to 20% of protein. So we'd want to broaden out the potential impact, but start first on that well-defined subgroup.
Yeah. Are you, I guess, how are you anticipating the effect in the different subgroups changing?
Yeah, well, I think the early data from SPG 601, we have a very, very robust effect in these zero and trace protein males. They are phenotypically severely affected with significant intellectual disability, very high anxiety, a lot of ADHD symptoms. They're patients that are requiring lifelong adult support and supervised housing. The very, very small percentage of them live even semi-independently. So I'm expecting a big potential improvement in the adaptive behavior and the functional outcomes once we're able to bring these brain circuits online and improve things. It may be differences such as an individual that, had to be picked up in a van every day is now able to ride public transportation, or someone that could only, sit in a day program and watch television can now have supported employment, or somebody that had supportive employment moves to independent employment. Those are the real outcomes I think that are on the cusp, are feasible, and that we really want to test for with chronic long-term dosing. That would make a big difference, not only in the quality of life of the individual and the family, but it's a huge financial cost to the world. I mean, almost all of my patients, my males, they have at least one parent or caregiver that's not able to have normal employment. They take care of their child, and they have to do that as long as they physically can. And then they worry about, as they age, who's going to help next. So those are the kind of consequences of the natural history we see now of Fragile X. But I think we're poised for a breakthrough, and we're excited about it.
Yeah, certainly it'd be a remarkable impact on this patient population. Tell me a little bit more about the kind of chronic dosing and and how you anticipate that. Kind of playing out therapeutically, I guess, for the individuals. I'm starting to kind of bridge this story around synapse formation, and is there continuous synapse formation that's required? I'm trying to understand how this kind of plays out.
Yeah, I mean, I think, specifically with the Fragile X example, we did some initial single dose work and we saw these bold effects, meaning that synaptic function was normalizing quite briskly. I think the one thing you worry about are things like tachyphylaxis and things wearing out over time, but that's where the specific SPG 601 molecule has chronic use preclinical data showing that that's not an issue that benefits preclinically continue. And it has other human safety and repeated dosing data, at least out to 28 days, showing that, that kind of tachyphylaxis or wearing out effect, which we've had with other molecules in Fragile X, in particular mGluR5 antagonists, where they had a real trouble with tolerance development. The BK channel modulator SPG 601 has no track record of that. So I think that is makes us feel good. And a brain that was able to immediately in our first trial have improved computer performance and normalized EEG signals, we really think is a brain that then is able to learn and communicate differently because there's so much interference in Fragile X. We know that there's excessive high frequency gamma band activity that really relates to degree of cognitive impairment and severity of anxiety. And with a single dose of the Spinogenix drug in Fragile X, that completely normalized. I mean, that's the strongest effect we've ever seen. So if we can keep that up, that's then a brain that is just able to process information differently. We know that when the brain responds to sounds in Fragile X, the responses are really abnormal. They're not able to pick up frequency changes, process language normally. So those are all things with chronic dosing we really want to track and see if the brain's response to its external surroundings can normalize over time. So that's really what we're looking for with the chronic dosing.
Yeah, one of the things you said really caught me, which is like this idea that a single dose has such a marked effect on synaptic formation. How do you think about like diminishing returns? I guess like are there ceilings to this? Is there like is there a threshold at which it's too much? Like how do you think about that?
That's a great question. That's where I look with the BK channel modulation in Fragile X with the immediate kind of effect. I look at the multiple sclerosis data, the human chronic safety data, and that came with the molecule. And there's no evidence of tolerance or tachyphylaxis because I do worry about that
Mm,
because we did
yeah.
experience that with mGluR5 antagonists. There's work supported by the Fraxa Research Foundation that preclinically validated this mechanism and looked at the molecule in animal models of Fragile X with that specific question, is there going to be tolerance? Is there going to be tachyphylaxis? We've been burned in the past. Can you reach this threshold? Is there a U-shaped curve of diminishing returns? And the molecule has not exhibited that. And that's using the same assays that, for example, showed that chronic mGluR5 antagonist dosing did have diminishing returns. So I think we're a bit de-risked there, but we're going to have to do the chronic clinical dosing studies to get those answers. But there's no evidence so far, preclinically or clinically, of tolerance, tachyphylaxis, and a loss of returns there. So we hope that the benefit we see, and we believe it'll be maintained.
Yeah, fascinating. And Craig, feel free to pass on this question, but I'd be curious to hear any of that kind of color on the other disease categories around ALS, schizophrenia. I'm curious, how are you seeing that impact with those patient populations?
Yeah, I think with those patients, so the first trials that have been done have been in ALS, and I think that we've used EEG as a marker supporting that work and see some really solid EEG target engagement briskly with the SPG 302 molecule within the first 4 weeks during placebo-controlled treatment, and we see those EEG changes maintaining over a 6-month period, which is really exciting. We've not analyzed EEG or a lot of clinical data from the ALS or the schizophrenia programs yet, but the same quantitative markers are there to be able to do the analysis.
Yeah. Um, how do you think about prioritization in any given kind of conversation with biotech and development? There's this kind of a conversation around prioritizing which which indications maybe go first or which programs go first? I'm curious, how are you thinking about that?
Well, first of all, I'm an academic scientist, so I don't think a lot about these business decisions about priorities. We want to help everybody, as quick as we can. But I think Spinogenix is well positioned to move forward the Fragile X work with the 601 molecule and the 302 in parallel. I think with 302 having the ALS and the Alzheimer's and the schizophrenia, some decisions will need to be made, along the way of resource allocation. But I think you follow the data and you look at those results and you make those decisions from there. So that would be my advice to do. But you need to have the data and the information from the multiple indications to decide what to prioritize. Because you're right, you have to make decisions, right? I mean, no one has endless resources in what you could do. But I think the fascinating and really novel part here is that the 302 mechanism and the tags approach is so broadly applicable to brain disorders in a really logical way. So I haven't seen anything come around like that in my career. I think that's in particular very exciting.
Yeah, incredibly exciting work. If we look into the future, maybe talk to me about how you envision combining the synapse restoration techniques with maybe some of the other programs that maybe you're excited about that are kind of on the horizon too.
It's a really great question. I mean, I'll give an exemplar where we have a little granular detail in Fragile X syndrome, right? So right now in Fragile X, there are phase 3 studies of phosphodiesterase type 4 inhibitors, currently sponsored by Shionogi. So they're focused on cognitive enhancement with computer-based testing. The electrophysiology data from that is relatively negative. They don't have that bold effect on excessive high-frequency activity like SPG 601, the BK channel modulator. So there's clearly different neurophysiologic effects of those medications. So from my perspective, if the Shionogi phosphodiesterase inhibitor were to be potentially approved, something like a BK channel modulator can be very synergistic with that. It has a different brain profile of response. I think there is rational combined pharmacotherapy. But again, I say that as an academic scientist, right? I mean, that has nothing to do with how you would ever join together mechanisms across companies and portfolios. But I think there are some rational combinations there. But I think using something very continuous as a quantitative measure like EEG can help you make some of those decisions. Like this is having this EEG effect and this cognitive effect, and that's synergistic or complementary to another molecule's effect in the same population. I think that's a much more logical way to put the pieces together versus, hey, here's 2 approved safe drugs, or here's 2 approved drugs in development that are different. I think now we can do that with a little more sophistication.
Absolutely. Um, uh, j- in closing here, I've got a few questions for you. Just continue to look into the future. So for the patients, caregivers, clinicians, investigators, and investors that are listening, what should they know or watch for on the horizon and be excited about as far as the Spinogenix programs go?
Well, I think Spinogenix and Fragile X, they want to look to the near term chronic dosing work and first really focusing on this male population and then expanding. And I think it's the number one preclinically validated and really the only mechanism with human positive target engagement data out there to get a lot of attention in the near term. With the work with 302 in ALS, Alzheimer's, and schizophrenia, there's going to be some long-term treatment data coming out in the near term that I think Spinogenix and I are excited about. And I think the sky's the limit for that concept. And I think, Spinogenix is first in the space of, altering synaptic spine morphology, as we said earlier, in ways that, can really restore brain function and restore it in a way that's quantifiable. So I think that is very exciting. And again, which disorder do you pick first with 302 is to be determined, but it is broadly applicable. I'm a psychiatrist. If we had non-antipsychotic approaches to schizophrenia that could restore, positive and negative symptoms and potentially cognition, I mean, that's massively game changing. And we know how devastating ALS is and Alzheimer's and the minimal effects of available pharmacotherapy. So I think there's a lot of excitement to follow there. I think the stakeholders are really excited about it. And I think Spinogenix has really embraced the stakeholder voice and been very open to extended open label treatment periods and thinking about EAPs and expanding access very early with their development program. So I really commend them for that and understanding that individuals with these devastating diseases, drug approval is really slow. And we
Yeah.
all know that. And are there ways to sort of to reach a common ground? And they're doing that. And I think that's really important.
Yeah. And maybe as a closing thought, outside of the synaptic restoration work, like this kind of category, what other innovations are you most excited about in terms of psychiatry and neurology, the kind of research that's happening in CNS?
I'm excited about gene and cellular therapy options, AAV mediated CRISPR gene editing strategies, cell penetrating protein work. Once we saw spinal muscular atrophy effectively potentially treated with AAV based gene therapy as a neurodevelopmental guide, that gets you really excited to think about technology and bringing it in to other areas. That's going to be most applicable to rare genetic disorders, but we're very excited about those transformative gene and cellular therapies that are likely going to require, very early intervention during the course. So I think that I really see those in partnership with small molecules long term for the population because, for example, Fragile X, they have a normal lifespan. There are a lot more adults out there than kids with Fragile X syndrome. So if we can bring a gene therapy to neonates, great, down the line, but we still need to offer effective strategies to improve quality of life for all the other patients who have lived for many decades with Fragile X. So I think there's really a dual small molecule and gene and cellular therapy role in these disorders, but I have excitement there too.
Yeah, certainly the outlook is remarkable and exciting, given some of this progress that's being made. And I appreciate the kind of leadership on your end and helping us kind of break new ground here. Craig, is there anything we haven't talked about today that you want to make sure we kind of we air out?
No, I think we hit it all. We even touched upon the reason I do what I do, and we're driven every day by clinical outcomes. I always tell people I have a Rolodex in my head of all the patients that I've seen, and that's why we do the work. And you understand the challenging outcomes, and you're driven every day to pound the door down. And that's why I get involved with Spinogenix, because they have a novel, logical approach. They're willing to be highly quantitative as a small biotech, right? Do things that big pharma's often not doing. And that's what I'm super excited about.
Yeah, incredible and inspiring. Well, Craig, thank you so much for taking the time. I've so enjoyed this conversation and I've learned a tremendous amount.
Awesome. You have a great rest of the day. Thank you very much.